All right. Well, thanks, Barb, and thanks, Mike, obviously, for inviting me again. You know, I think we're supposed to talk about who we are, yeah. So I was an academic, like many of you, for a long time. I was a professor at George Washington University at a lab at the Red Cross. My background is in vascular biology, extracellular matrix biology, and happily successful, at that time, reasonably successful academic, and just tooling along. And then something happened where I was asked by Novartis to join a gene therapy company that was a subsidiary of Novartis. So I thought, well, you know, I had some ideas. It would be a great way to translate some of my academic research into an applied research environment. And I did that for 2-3 years, and I think my wife was going to kill me because I had 2 full-time jobs, basically. So I moved over to Novartis, and my background is really varied. I mean, I've done a lot of small molecule, particularly in the hypertension space, and also monoclonal antibodies. When I was at Novartis, my group was the first to start the PCSK9 program there. So a varied background. I then continued my journey in pharma with Pfizer, a little bit at Shire, and then the last probably, I guess, 6-7 years, mostly in a small company environment. I first joined a genome editing company called Precision Biosciences, which use homing endonucleases, which probably is a genome editing system you've never heard of, but it's actually a great platform. And I headed up the gene therapy world there. And then for 4 years, I was at Stoke Therapeutics. You're going to hear Adrian Kramer's great story on SMA, SMN. And he was one of the co-founders of that company. So I headed up preclinical research and development there for 4 years. And I'll show you a little bit of that data. Then I recently I started a new company called with some colleagues in the circular RNA space. But most recently, I am semi-retired, and we just got back from a month in New Zealand. So I'm very relaxed and well. In terms of my job, you know, the first time I was invited to come, I think I had a board conflict. I couldn't make a couple of days, so I think I blew in, gave my classic Stoke talk, and spoke to some folks, and left. And I've since done a couple more of these, and I realized that most of you guys are in the small molecule and world and in other spaces. So I think my job is a little different. I've taken a lot of the data-rich stuff out because it's not particularly relevant to what most of you are doing. I think my job right now is going to be 2 things. So, you know, it's lunch, and you know, pretty soon you'll have the postprandial stuff, and you're going to be maybe a little sleepy or whatever. So I want to give two key messages. One is that the RNA field is a place to do drug discovery. There's lots and lots of targets, and I will show you some details of that and why. It's a burgeoning area, and it's a place that you really should think about when you're thinking about your target, you're thinking about your approach to a disease, to also consider that area as well. And then obviously, because of my background, I'll tell you a lot about the ASO biology, the things that we've solved, the things that we haven't, you know, that's still a problem, and we'll go forward on that as well. So hopefully it'll be fun to kind of learn something that's a little different, and maybe give you some new ideas as well. Okay. So yeah, you really have a hard time seeing this. Okay. All right. So DNA is transcribed. It goes to pre-message, which is then spliced to message, which then is translated to protein. And everybody is focused on here, right? And I took a quick look, and pretty much most of you are focused on small molecules. There's then a small group that's focused in the gene therapy space, and then I hear maybe a few people that's interested in the ASOs, and I think that the point of this slide is simple. Is that when you look at this process, an ASO, an oligonucleotide, which can be with high fidelity, bind to a cognate sequence, allows you to really very specifically, in a precision medicine way, look at very specific targets, specific genes, and you can interfere with the transcriptional start sites, the translational start sites, the RNA splicing itself, and we'll talk more about that, as well as obviously the process of translation to a protein product itself. So, since pre-mRNA splicing was discovered in 1977, and 78, a group started looking at the idea of modulating that. But it actually took 20 years before the first oligonucleotide-based product was approved by the FDA. Since then, there's been now a small handful of those, and like I said, you hear from Adrian the lovely story of Spinraza, as well as some of the other research that he's engaged in. But it's a slow process. Why is that? Well, I think you've been hearing some great talks about drugs and the feasibility of drugs and the size and the Kapinski Rule of 5 and so on and so forth. Well, if you look at an ASO, it's about 10 kD. It's highly negatively charged. And also, your cell does not like to have oligonucleotides floating around because those are usually a bad signal. So it's degraded very easily. So those are the problems that the field had to encounter, that had to solve as it moved forward in this whole process. And we'll talk through about some of those problems. And let's see. So I won't be talking about RNAi, but RNA interference, some of the key elements and the thought processes are pretty similar to the Gapmer concept that I will talk about with antisense oligonucleotides, double-stranded obviously, but the ability to really get an effective RNAi to work is very similar in terms of how you want to design your ASOs, the kind of substitutions, and so on and so forth. So very, very similar lesson, if you will. The Gapmers are the ones that allow degradation of specific targets, where you have a RNA, DNA, RNA structure, and that triggers RNase-H mediated degradation. RNase-H is the enzyme normally used for replication and repair, and it recognizes this and degrades the target. What we'll mostly talk about are the steric blockers. I think these are, and they could be uniformly modified or mixed modified, and we'll again discuss this in greater detail. But the point is that there's an incredible amount of diversity in terms of the ability to using steric blockers, and I'll discuss this. So the topic for today is, well, I'll spend a good bit of time telling you why RNA-based therapy and how does it work. We'll then talk about the hurdles for ASOs, the drug-like property I just talked about, and the delivery issues, which is a big issue. And again, you heard a very nice talk on ADME earlier today, and we'll discuss that in the context of ASOs. Toxicity as well. I'll spend a little bit of time. I stole one slide from my better half, who will tell you all about the toxicology components of drugs. And then we'll kind of switch a little bit and talk about some other approaches. In particular, I'll give you a small example or a taste of the vectorization of ASOs that allow you to take the same targets, but now do it in a very different way. And finally, a few future comments. So the application of antisense oligonucleotides, as I talked about with the gapmers, it's pretty clear, and that's on the left side of the screen. I don't really need to go into the details of that process, and as I said, what I want to talk about are the steric blockers, and where again you can inhibit a variety of parameters, such as the splicing effects, which we'll talk a lot about, but also upstream open reading frames, translations, initiations. Also, as you know, there are a lot of multiple poly-A sites, which also drives diversity, and you can also modulate that, as well as microRNAs and long non-coding RNAs, which we'll discuss a little bit as well. So, I think we must have. Oh, I see. This was well, anyways, that's okay. So I think one of the so I think we're missing some things here for some reason. I'm missing a figure which shows you about all the different types of splicing that's available. So you have to visualize that. So RNA splicing is complex. It's what really drives the diversity that makes human a human, and a mouse, and a fruit fly, right? It's really the big component of that is splicing. So you have alternative splicing where exons are included or excluded. You have alternative 5' and 3' splicing junctions that it's incorporating. You've got exotrons, which are actually introns, which can occur within an exon itself. And I also talked to you about poly-A sites and so on and so forth. So that provides a diversity, but also it drives the diseases as well. It turns out that probably 15 or 20% of human genetic diseases are caused by splicing errors. Now, splicing effects can be cis or trans. Cis effects, I think there was an example that talked about the CFTR disease, cystic fibrosis, right? So it turns out that probably a quarter of that is due to splicing errors, essentially. And you can have trans as well, and this would be SMN is a perfect example. DMD is a cis example. Myotonic dystrophy, muscle blind is an example again of a trans factor, because those are all factors that are associated with your ability to actually perform appropriate splicing, essentially. So that's one thing, and there's a very large proportion of splicing errors that drive genetic diseases to take home. Okay, so then the other thing that I need to tell you about is nonsense-mediated decay. As probably most of you know, there's an exquisite system in which your body checks the fidelity of the messages themselves, because obviously translation of messages that are inappropriate is very bad for the cell's health and well-being. So in this case, what happens is that there's a scar for each of the splicing events that occur within a gene. And that's then read as part of the initial translation process itself. And when you have a translation that incorporates some of these different exons and so on and so forth, you have often what's called a premature termination codon, PTC. So that stop codon, when it occurs early on, and in this fidelity system, it evaluates this, and it sees that stop codon, and then it sees one of these junctions. It says, uh-oh, there's something wrong with this message, and that message is degraded. So at Stoke Therapeutics, where I headed R&D for 4 years, this is what we took advantage of. This was a very strong. There was a very strong bioinformatics group. Well, obviously, did a lot of RNA-seq data. We collected RNA-seq data from a variety of other places as well. And from all of that data, what we were able to do, and these numbers, this is from a published paper that our lab published, but the numbers themselves are not exact because they're changing all the time as new data come and go. But the main point is that there are thousands of these events where the kind of splicing changes I told you about that actually drives the generation of a premature termination codon, either within that exon itself or because your reading frame has changed in an exon downstream, right? So then what happens is that you drive the NMD events. And as you can see, there are thousands of them that are specifically associated with diseases, and these are all the different kinds of alternatives that we're talking about. Okay. So we thought certainly one of the things that made a lot of sense was to go after haploinsufficiency diseases. What haploinsufficiency diseases are is where one allele has a mutation and you have loss of expression. And just by having 50% expression of that particular gene, you end up with a disease. And that's particularly prominent in the brain because I think because of the way the organization of regulation and other activities, you end up with these exquisite events where the level of a particular protein product has to be at a certain level or there's a problem. So with the Stoke Tango method, that's a targeted augmentation of nuclear gene output, and Tango because Adrian and one of the young co-founders at the company were from Uruguay. So in Tango supposedly was a invention of Uruguay, and that's why we got Tango ASO. But anyways, the idea is that when you have a mutation on that right-hand side, this red side, obviously what happens is that messages are made, there's a missense or there's a nonsense mutation, and you do not have a productive protein product. If within this gene there's also a Tango event, if you will. Now what we know then is that you have two of these activities. In this one case, because you have that NMD event, it's still non-productive. So you only have this productive event. And in the context of, obviously, the mutated gene, it doesn't really matter because it's not making a protein anyway. So what happens is we can alter that. We change that event and drive the non-productive message into a productive pool itself. So in the context of the mutated gene itself, nothing changes because all you're doing is now making a productive message that has that mutation from the disease, right? So you're still not making any protein. But on the other side now, that non-productive event now is now productive. So what you're then driving is you're compensating for the haploinsufficient product. So, and this was published a couple of years ago from our group, and the idea is that to show you that these are broad-based events. So what you're looking at is in the presence or absence of our oligonucleotide in increasing concentrations, probably a little bit too small for you to see. And what you see is then is the decrease in non-productive event, an increase in the productive message, and an increase in the productive protein. And I want to use this slide to really show a couple of things. One is obviously that for haploinsufficiency disease, in this case SYNGAP1, a genetic epilepsy, that's great. But also you can do it in terms of other diseases as well, even if it's autosomal recessive, for example. as long as that there's activity, there's product in that protein, you can still do the same thing. You see the same patterns, and then obviously pathways as well. In this case, it's PD-L1. In the context of thinking about autoimmune type of diseases, we can actually upregulate PD-L1 as well. So, like I said, I took a lot of the old data out, but the bottom line is that we focused on Dravet syndrome, a genetic epileptic disease, which is really quite devastating. It's a childhood disease that the onset begins perhaps at 6 months to a year, and you have seizures that are observed. So on the left-hand side is our Science Translational Medicine paper, which has a tremendous amount of data in it. But I won't bore you with that data, but I just want to show you this one particular slide, which demonstrates that, and this is now what we're looking at, is the ability of the ASOs to impact on the expression of SCN1A. SCN1A encodes, it's a sodium channel gene that encodes NaV1.1, and NaV1.1 is crucial for the function, particularly of the inhibitory interneurons, the PV interneurons. And so in this particular case, what you're looking at is an animal model that has lost one allele, and you can see that this is the expression of NaV1.1. And so what we know about NaV1.1 is that it's expressed very low at birth, and then there's an increase in expression over time in early childhood. And it's this requirement as part of brain development that's missing. And so while there's substantial NaV1.1 expression, it's insufficient and causes the disease. And so what you can see is that when we add our ASO, what we've done now is we've left shifted that profile, and we've increased the expression. And not on here, obviously, is the wild type, but basically it really overlaps with the wild type. And on the right-hand side is a different paper, and this is work, and we've done collaborations. This is collaboration with Laurie Isom's lab at Michigan, and it was really a beautiful collaboration, and hopefully a lifelong friendship as well. On the right-hand side is a different paper that we worked on with a group at UVA, and what you're looking at here is seizure frequencies. What we were looking at there is specifically the interneurons, their firing, their activities, and there was a lot of data associated with that. But what you can see is that when we headset these animals, and this is like 15 to 20 on the left side, days postnatal and 20 to 40 days postnatal, there's this incredible ability of our drug, STK-001, to inhibit the seizure frequency, essentially completely inhibiting here and then showing a very dramatic inhibition of the seizures. So we generate a tremendous amount of preclinical data, and we're able to push this into the clinic. I'm obviously no longer part of Stoke, but this is the latest highlights from the end of 2022. And so you can see that there's positive first-line data. We've been able to show that the drug developed added intrathecally, is well tolerated all the way up to 45 milligrams. There was a 50% reduction in baseline convulsive seizure frequency and in the majority of the patients, and a greater than 50% reduction in seizure in 4 out of 6 patients. And this seizure frequency reduction is really consistent with what we anticipate to be happening, because what goes on, obviously, is it takes a while for the drug to drive the increase in the level of NaV1.1. But one of the things about these diseases that's really pretty amazing is that, there are, I think as Mike indicated, a number of very good anti-seizure medications. But often with these genetic epilepsies is that there are a lot of other parameters, factors are going into the system as well. So meaning that not only do they have seizures, but they have developmental abnormalities. They've got a variety of problems with respect to their eating disorders, sleeping disorders. They have a loss of ambulation in a variety of settings. And so these are just devastating diseases for these kids, their families. And so we are starting to see signs that some of these comorbidities may be trending in the right direction as well. And as as a scientist who's been in the business for 30 years or so, it's really gratifying to see something like this. And I think I was talking to some folks at one of the tables. It's you don't get that opportunity too often. I've generated lots and lots of wonderful preclinical data, and in a variety of different settings. But have that preclinical data and to have it to move forward and translate into the clinic is something that's really pretty amazing feeling. And I hope that all of you have a chance to experience that at some point. All right, so this is the feel-good part. Now we're going to move into sort of what are some of the issues and the problems. So we're going to talk about drug-like properties and delivery to target tissues. So the first element obviously is the modifications that we were talking about. So you can see they really fall into some different categories. One is the changes that were made with respect to the ability of the ASOs to withstand nuclease digestion, as well as having more drug-like properties. So the first part is that it turns out additions of phosphorothioates, adding a sulfur group here was very critical to enhance the stability of the drugs. The second element is the modifications, and the methoxyethyls actually is one that we now use routinely. And again, these are modifications. 2'-methyls as well florals also have important applications. And these are modifications which then allow one to both have differences in affinity and stability as well. Now, some of you may have heard of the locked nucleic acids, or the CETs and the tcDNAs. These lock the nucleic acids into a certain conformation, and that really drives the binding of the RNA to its cognate target as well. But the problem with some of these is that when you have too many of these, actually they cause perhaps more problems than they're worth, and you have to be very careful in terms of the incorporation of these. And then finally, there are other alternative chemistries. PMOs you probably have heard of, all the drugs that were talked about earlier. And by the way, I'm really happy that some of the talks that I'm hearing now have a little bit of ASOs in them as well. So that's really nice to see. And PMOs are the drugs that were used for the initial approvals for some of the DMD applications. So the take home, well, stability, affinity, lipophilicity, protein binding properties, and stereochemistry now can be successfully manipulated in the field after these 20, 30 years or so. As I indicated, the MOPS is the standard that we use at Stoke Therapeutics. And then what you can do is mix and match and screen for the right properties. One of the advantages, so ASOs are regulated as small molecules. Certainly one of the advantages that when you think about ASOs in the biology that we work in this field is that these screens are actually, we do walks through specific regions, we're probably talking about maybe tens and hundreds of molecules, not thousands and perhaps hundreds of thousands of molecules when you do our screens. So you can very quickly determine whether something is going to work or not. Once you find a lead, if you will, or a hit, you can then match, and sometimes you maybe want to increase or decrease affinity. You can use some of the chemistries I told you a little bit about to do that. There are times when ASOs do self-aggregate, and so you have to you can change that as well, and obviously cellular uptake. Now, this is my feeling from looking at the literature. Others, I think, may perhaps feel differently, but bottom line is that I think that additional direct nucleotide modification to address PK biodistribution and endosomal escape is really very difficult. I think they're just intrinsic issues that seems to prevent you from making improvements in that area. But there's still certainly lots of people that are trying. And I think most of the efforts have moved to conjugation and encapsulation to address these issues. So, you've heard this very nice ADME talk. So distribution is one of the key hurdles going forward in oligo space, basically. And obviously you want to get these are precision medicine tools. So if you can get it to the right place, it's going to work. So the question is, how do you do that? So getting into the right tissue, getting into the right cell types, that's the goal. Now, direct administration in the compartment of interest is something that has actually worked very well. For the eye, my team were able to put a second program that's moving into the clinic, and for the eye, it's for autosomal dominant optic atrophy, and for the eye, it was very straightforward. We were able to directly inject into the eye. I know, I saw that. That's okay, but it's actually done all the time. For the anti-VEGF therapies, I think that Mike had talked about, and so very easy. You get very nice broad distribution, and we get very very nice activity. For Dravet, actually, we do intrathecal delivery again, directly into the compartment. But obviously, there, I think the risk-benefit ratio becomes a real problem. In the case of Dravet, no, but perhaps in other CNS diseases, injections 3 times a year, lumbar punctures may not be something that people really want to do for a long time. So this is where we'll talk about other approaches that one can potentially take. All right, so conjugation is really the key. We'll talk a little. GalNAc is something that has been perfected initially in the RNAi field and has been shown to be very effective, and also now in the ASO gapmer space as well. So the liver and the eye are 2 really easy places. The brain we'll talk about specifically in a little bit. Lipid conjugations and peptide antibody conjugations are the things that are really what people are working on, and then obviously encapsulation as well. So this is kind of a nice summary of some of the different approaches. I think there was some discussion before, I believe, about cell-penetrating peptides already, but these are some of the passive targeting, if you will. A lot of work over the years. I have to say, without a lot of incredible successes. I think that, great proof of concept, movement forward, preclinically, even in preclinical animal models, but in the context of moving things into the clinic, I think not quite as successful. Because the fatty acids, for example, are slight distribution changes, and they're just not enough. The active targeting, and we'll talk a little bit about that, Guy already talked about the transferrin receptor as potentially one for the cross the blood-brain barrier. I know that at Genentech, I worked on that for quite a bit, as well as other companies. But for the muscle, it looks like it's working pretty well, and we'll discuss that briefly. So obviously, lipid nanoparticles, LNPs, some limited success, and then obviously polymers and others as well. So there's a lot of effort and a lot of work in this space. Just a quick note about the transferrin receptor. Both Avidity and Dyne are using that, conjugating into RNAi or ASOs, and that is moving forward. And Avidity recently posted positive interim data from their study for AOC 101 this is DMPK reduction, and they show splicing improvements and early signs of clinical activity after dosing with the ASO. So you're mostly interested in the brain, the CNS, and as I indicated, really from a ASO perspective, because of some of the size and other issues and charge issues associated with ASOs, it's very difficult in terms of systemic delivery at this point. Although it's a very active area of research. As I indicated, we do intrathecal delivery, and we've been able to demonstrate very nice biodistribution of our ASOs in different parts of the brain. There's intraventricular delivery. There are other delivery processes as well. And there's, from a tool perspective and the ability to do this in a clinical environment, for infrequent or maybe once in a lifetime type of delivery, it's very feasible in terms of direct injections into the brain, and there's a lot of work in that space. IT delivery is really not that terrible, and it's long, but the frequency is the question. Probably many of you maybe have had an IT lumbar puncture. I have. It's just, not that bad.
I actually have a question about the intraventricular. Do they need to drill?
Yeah, but it's stereotactic, and they know exactly how to deliver the drug, and it depends on how you do it. There's even now this idea of sort of setting it up as a kind of a almost like a depot kind of environment. So you have it set up, and they've set it up, and now it's there, and then you can yeah, right.
It's like deep brain simulation.
Yeah.
It would wire directly to the subcellular area. Yeah.
Exactly. So there's a lot of advances in that space as well. Yeah, very, very, very interesting, very exciting. And this is a slide from Frank Bennett from a 2018 course, the year before I joined, and basically it's just to show, this is using a target MALI1, that you can get broad distribution, and like I said, we've now shown have the same data, and I think I've published that in different abstracts and talks as well. So bottom line, delivery is the key, and right now there's for the CNS, because it is an area where there's a lot of interest from the ASO community. Just kind of give you an example. There's a very large collaboration between Ionis and Bicycle Therapeutics, as well as one between Alnylam and PeptiDream that's focused on the CNS delivery process. Obviously, the entry and decloaking within a cell to get to the right place is also a very important component. All right, the other part is just very quickly, I'll go through some of the tox-related issue as well. And this is a slide I stole from my better half. And here, the main thing I want to point out is a couple things. Is that ASOs are somewhat similar to monoclonal antibodies that I talked about. there's a set of class effects that you can really look at and address relatively early and think about. So if you're looking at ASOs, probably one would want to move up and think about a couple of things. One of the first things is that it's easy to do in vivo tolerability screens, and we do that. And I know that some of the other companies do that on a routine basis. And then you look for things like complement activation, thrombocytopenia, inflammation. Those are things that are known that with high doses of ASO in the systemic compartment that you will potentially see. But in terms of gene tox and other kind of things, I would really put that at the back end to do only what's absolutely required by the regulatory agency, because really the field has not seen any of that with the oligo-based therapies. So I think that's the main thing I wanted to point out. And then just kind of a very brief summary. So there are sequence-dependent, non-specific primary pro-inflammatory effects. So CpG runs are clearly pro-inflammatory. G runs are not good. Complement activation is certainly a part of the driver for that. So you can really screen that out early when you're targeting and designing your oligonucleotides. Hybridization, independent liver and kidney toxicity due to accumulation. So ASOs systemically delivered primarily end up in the kidney and the liver, and depending on the chemistry, can be more in one versus the other. The accumulation could be due to specific protein binding. So essentially, there could be a possibility that these sequences function as aptamers, basically. And in terms of kind of going back to the kind of the PK component, is that you even when you inject the ASOs into the CNS compartment, it really very rapidly goes out into the systemic environment, basically. So you can evaluate the PK in the blood even with the IT delivery. Thrombocytopenia is a sequence-dependent but hybridization-independent phenomenon. There's a small percentage of ASOs that have those activities, and it's much more severe in non-human primates than in humans. And then there's hybridization-dependent off-target effects. Obviously, early when you are doing your analysis, trying to find your target, the bioinformatic component is really important, looking for off-target sequences and then deciding whether those off-target sequences are worthwhile to evaluate or not. So, so that's the ASO piece. I'm going to briefly talk about the vectorization component and then finish off with a few comments. So the vectorization component that I'll talk about, which is very much been around, basically, I think late 90s, early 2000s, really, groups have already started thinking about this. And the idea is to use the U7 small nuclear RNAs. And that normally function in histone activity. And what was done was to remove that histone recognition component and change that into the recognition of whatever your target your ASO target gene is, essentially. So what you've done is that you build a platform that presents the ASO in a gene therapy environment. You can now incorporate these U7s into an AAV. You can put multiple copies of this, and now you have a gene therapy system that uses the oligonucleotide or antisense principle to drive all the changes that we've talked about, whether you want to increase a target gene, decrease, change the profile of that in terms of the particular protein you want to make, and so on and so forth. So this is just a little bit of data so on the left-hand side, you see our gels, and the right-hand side. This is a really beautiful story in a lot of ways. So now this is another a DMD program, Kevin Flanagan's group at Children's, and they essentially, so there's a exon 2 duplication that goes on in a small percentage of DMD patients. And what the biology that was found out by Kevin's lab and others was that it turns out that there was some clinical data that indicated that for some reason, if you delete the duplication, that the patient actually now, although it still has the mutation, I think in this case it was only one of them was deleted, and you have this NMD event downstream that prevents the production of dystrophin, that the patient was fine. And it turns out what happened is that there's an iris, a internal start site, basically, an iris site that is then revealed, that then drives the expression of dystrophin itself. So what they did was then developed an AV9-based exon skipping system, skipped exon 2, and then they looked at what happened. And in this case, in the animal model, you can see the gel on the left-hand side, but probably easier for you guys to see is on the right-hand side, is that with increasing doses of AAV, you can see the restoration to almost wild type of the dystrophin. And this dystrophin is not like Becker's because it's really a dystrophin that's pretty close to fully functioning, even though it's missing that initial small component of dystrophin. So a series of papers were published by his lab and by the group, and demonstrating, again, safety and other components. And then at the ASGCT, he presented the data, or his colleague, Megan Waldrop, presented the data regarding the first clinical trials of using U7 snRNA gene therapy. This phase 1/2 trial looked to be quite exciting and positive. 3 patients treated, well tolerated, and clear demonstration of increasing dystrophin positive fibers. So the point is that when you look at this huge body of potential targets that you can manipulate in a variety of different settings, and that in the RNA world, there's a lot of different approaches. You can use oligonucleotides, and you can also use other approaches. In this case, what I've demonstrated for you is an AAV gene therapy-based U7. I can tell you that there are other approaches. I recently started and left a company that use a very different approach using circular RNAs, and we're thinking about it in terms of similar kinds of concepts as well in a variety of settings in this world. So let me just leave you with a couple of thoughts. Obviously, you've heard about Spinraza, and Adrian's going to tell you that story tomorrow night. And that was approved in 2016. And then Zolgensma was approved, and that really, a lot of that work really came out of Children's, where Kevin Flanagan was, and academic translation into a pharma situation. That was approved in 2019, and now there's a small molecule has been approved, Evrysdi. But initially that was PTC Therapeutics, PTC, and then that was a partner with Roche, so proof of concept now has been shown for a RNA target with small molecule approaches, gene therapy approaches, and obviously ASO. So my point is that when you are looking at this, all of these should be considered. I would say that ASOs is still the most credentialed, and when I first got here, I was chatting with someone, and I think the lady told me, she says, yeah, Spinraza is kind of taken over by Zolgensma. I don't fully agree with that. Actually, it's really interesting in what's playing out in this world. And, each one of them have their pluses and minuses. Quick onset with respect to Spinraza, the direct impact. When you have a baby that's born and has SMA, you probably want to give that baby Spinraza and drive those positive events very, very quickly. The gene therapy, it's kind of like we talk about it. So, you have an effective drug, you're giving it by lumbar puncture every 2 months or 3 months or 4 months. This child becomes a teenager. He's playing basketball and, whatever. He's probably less interested in getting that lumbar puncture. So maybe a gene therapy. So I think they all have a place. And small molecule is, Evrysdi really has driven a lot of investment in that field. And there's a lot of interest, a lot of little companies. Adrian actually is co-founder of another company, I think, called Skyhawk. Maybe he'll tell you a little bit about that. I don't know. But I think that the proof is still in the pudding in that it's very difficult to target some of these places using small molecules as well. But obviously there's effort, and hopefully there will be successes. So when you think about all this, don't think, oh, RNA target ASO, I don't do ASO, I don't, whatever, right? But really think about it in terms of the biology of what's going on. So you've got all these different diseases, and you've had to think through about in the context, okay, is direct invasive delivery of my drug, is that reasonable for this indication? What's the risk benefit for the patients? Do I need a systemic delivery really? Then I may need a small molecule or whatever, or maybe some AAV. There's a lot of effort in there to drive systemic delivery AAV. Encapsulation of ASOs is also ongoing. I told you about some of the conjugates as well. So all of that is possible, but do I need that? And what's the most likely approach for me to drive that process? And what's that risk benefit? So your choice of approach really depends on that biology and on the nature of the disease itself. So I will just leave you with one last thought, which is that RNA sciences continue. I mean, even as, for this talk, I decided that maybe I need to look at what's going on and kind of polish up a little bit and think about where things are. But really, the field is continuing to grow with long non-coding RNAs. People have been trying to target that. There's alternative biology associated with that. MicroRNAs, that's an ability to really manage and regulate a large number of targets. Backsplicing, I told you, I've been some work in the circular RNA space. There's transsplicing as well, you can see here, where, and that could be utilized possibly in the future also. And then there's, interesting, I found this paper about, these microexons. There's actually been quite a bit of publications in this space. That these are ones that probably many of us, bioinformatics and other ways, are not actually seeing. They're starting to be seen now with the newer techniques in terms of analysis and therefore evaluated. So they're probably about 1% of alternative splices. However, it's 1/3 in the neuroregulated alternative splicing. And it appears to be highly conserved. So what's going on there? So there's a lot of biology, and I think definitely keep an eye on that, and hopefully you can find a way to approach your disease or your target via one of these methods as well. Thank you.
Thank you so much. I'm a child neurologist. I dose people with ASOs. So I have two questions. One is, how long do we think ASOs will be needed, right? And in kiddos we do have many patients who get the ASO for Spinraza from a young age. Do we have any concern about what's the long-term effect of ASOs on the body? And like, what is if and when do the antibodies kind of come out, and how will that affect kind of the field, recognizing that nusinersen's been in clinical trial for what, 15 years?
Yeah, so as far as so ASOs, as I said, as a small molecule set of drugs, appears to have a set of parameters that give you certain advantages and disadvantages. And the advantage, certainly one of the advantages, is that in terms of the byproducts or downstream products, there's very little. And if you think about it, the only thing really you're generating is N-1 and N-2, basically. So from a long-term safety perspective, there's not a lot that we, people in the field have seen so far. It's really more about the level of dosing, and as far as ADAs, I think Guy talked about, ASOs are notoriously difficult to generate antibodies against. And, when we're required by the FDA to set up some of these assays, so it takes an incredible amount of effort just to try to find something so that you can, have an assay to set up. So I think to be seen so far. There's not anything that there's been that I'm aware of that would suggest long-term worries. And certainly Spinraza has been approved since 2016. So I think there's a lot of clinical data associated with that. And your second question was? Okay.
Thanks. That was great. In terms of preclinical efficacy data, what are companies or the FDA looking for? Multiple models, multiple species, in vitro?
Well, from a efficacy perspective, it's interesting. I think, obviously, maybe you hear more from the regulatory perspective, but from my experience in overall in general is that obviously it depends on the reviewing committee and the people that are associated with it. The neuro division is pretty tough, I would say, and I would say rather, shall I say, traditional, at least some components of it. Different divisions may be different. And so there's a substantial requirement, and obviously there is a need to demonstrate efficacy. Obviously, in some cases, you may not have good in vivo models, and so that could be somewhat problematic. Ideally, if you have an in vivo model, you'll be asked and be required probably to use it. I don't think you need multiple models, no. And as far as in vitro to in vivo translation components, if you can demonstrate that, this is the best I can do. Now, one of the issues, and we had a fabulous finding in our system where, in fact, in both our programs, the ASO that we found that worked in the animal models from mouse into man, and with several stops in between, is that it was the same sequence. Now, often when you're focusing on sequences, it may not be, depending on the conservation. So that provides you the additional level of complexity, potentially. So you have to look at that, and that's where the choosing of the early on of the targets and where you want to be is obviously a very important component of things. Yeah.
You showed intrathecal injections. So how long these ASOs stay in the CSF, fully functional?
Yeah, so the sort of overall PK component is what happens is that you inject the ASO into the CNS, you get a PK profile that goes up, and then there's a long. I think you heard about the lag part into infinity. There's a very long lag in the CNS. And I think what happens is that in the brain compartment, there's a certain amount that gets into the brain. There could be places where there are sequestered in the ECM component, or they could be in the cells themselves. They're taken up, and these ASOs work in as a they could potentially work in multiple rounds, basically. So maybe what's happening then is that you have the CNS compartment, then the majority of it, a very large component, goes out into the systemic, and then it goes into the liver and into the kidney. But the part that's in the brain is there's a very slow release of it, and you'll see, and eventually this very long tail that will show up even in the systemic compartment as well. And so I think there's this dynamic of what's going on in terms of activities where it can maintain a relatively long level of activity, basically. I think, like I said, we think dosing every maybe 3 times a year is certainly a possibility. Yeah.
Hi. I have 2 questions regarding the RNAs. The first one is, how do they enter in the cell and do the endosomal escape, if you have any idea? And then the second one is, how do you know which nucleotides you have to modify and what modification? Because, okay, the chemistry is established, but then you have 20 different combinations that you can do. So that is a huge number at the end. So how do you select what would be the first, or if there is any rule of thumb to select any of those chemistries?
Yeah, really good questions, and I wish I knew all the answers, but I'll tell you what I know or think. With respect to the first question, it's a little surprising actually that the data on how ASOs get in and then are escape is really not that incredibly well understood. I think what is known though pretty clearly is that there are often, and this is where you can be fooled, that the ASOs may not be in the right compartment. So you may think you have quite a lot within the cell, but they're actually in a non-productive compartment within the cell, and it appears to just sit there. And whether that eventually gets released in some way or not, I don't know the answer to that. So there's some suggestions of some receptor-mediated uptake, but I would say that the literature is really not all that well developed, so you can say with great certainty. And obviously, depending on the chemistry, now that we have all these different chemistries, in terms of what that might be happening as well. The second part with respect to the screening, I think I can give you a more straightforward answer, which is that, as I said, we usually use the MOPSs, and in the context of some of many of the things that we're looking at, it seems to work pretty well. And so when you want to improve the affinity, or you want to try to change some of the parameters that we were talking about, then there's really no specific answer. What my chemistry team usually did was we would go through, for example, if we think that there's potential need, for example, for greater affinity, we might go through and take all the Gs and change them to florals, for example, and we screen that and we look at. So the idea is that you do screens of, I think, I don't know, I'm sorry, I forgot. I think there was a discussion about the small molecule screens in terms of what don't synthesize 100 molecules, do 10, look for what works, take the best, and then do. So it's a similar kind of concept. We do the first set of screens. If you get something that shows a positive, and then you do chemistry around that area, and you see whether that then leads you to something else as well. So it's quite similar to the small molecule approaches there. Yeah.
So I have a question about, I used in the past a few years, couple of years ago, this antisense called Vivomorpholino. Do you
The morpholinos?
Yes, yeah.
Yeah, PMOs.
That's right, yeah.
Yeah.
What do you think? I mean,
Well, they are wonderfully safe. They're like Teflons, right? I mean, and that's why you can give grams and grams of this stuff and nothing will happen. But that's the problem. Nothing happens. It just goes right through you and it goes right back out. And the reason they worked in DMD is because of the injuries to the muscle, presumably, the injury to the muscles, which left them open and available so that the ASOs can actually get in. PMOs actually have quite high affinity. So they can work well if you can get them into the cell type. So there's been a lot of work to try to target and to enhance that with what I think is called the PPMOs, with these peptides. Some of them I think are kind of like cell-penetrating peptides. The problem is that they then drive toxicity really, really high, and that's the unfortunate problem with that. Yeah. But PMOs are great for, proof of concepts and other kinds of things. Yeah, very safe.
Okay, let's thank Dr. Jean Liau for a wonderful presentation.